Okay, welcome everyone to this new video in cell biology. In the previous video, I talked about the structure and function of the cell membrane's carbs, what's known as the glycocalyx. In this video, I will be describing the synthesis and modification of the cell membrane's proteins. So first of all, let's talk about how proteins are generally synthesized inside a cell. So protein synthesis always starts in the cytoplasm using structures called ribosomes, or in this case, free ribosomes because they exist freely in the cytoplasm. They're not attached to the endoplasmic reticulum, for example. The process in which a protein is synthesized is referred to as translation. This process happens after another one called transcription, where a certain gene responsible for a specific protein is copied into another structure called messenger RNA or mRNA. The mRNA is like a document with instructions, or it's like a manuscript that is used by the ribosomes to produce these proteins. So to give you a simple example to visualize this process, it's like Google Translate. So if you go to Google Translate, you have a box where you enter the words that you do not understand, and then you have another box where you have a translation for the words that you understand. In the cellular case, or in the cellular standpoint, the ribosomes are like Google Translate, where the messenger RNA is like the words that the cell does not understand, and the proteins are the words that the cell understands. Okay, so hopefully this simple example actually makes this a little bit easier to understand. Protein synthesis can finish completely in the cytoplasm, or it can finish in the rough endoplasmic reticulum. So as I said before, protein synthesis always starts in the cytoplasm. It can be paused in the cytoplasm and then translocated to the rough endoplasmic reticulum, or it can actually finish completely in the cytoplasm. So it starts in the cytoplasm, but it can finish completely in the cytoplasm, or it can move to the endoplasmic reticulum to finish there. The process where protein synthesis is paused in the cytoplasm and then resumed in the ER is referred to as cotranslational translocation. Cotranslational because we're talking about two different organelles working on the translation process, in this case the ribosomes and the endoplasmic reticulum. And we have translocation because there is a change of location from the cytoplasm to the endoplasmic reticulum, or the membrane of the endoplasmic reticulum in this case. This is the most common process in eukaryotic cells. There is another process called posttranslational translocation, but it's not common in eukaryotic cells, so that's why we're not going to talk about it. But from the name, you could tell what this means. So after translation, there is a translocation. So after the protein is completely finished, the protein moves to the endoplasmic reticulum for further processing. Protein synthesis usually starts with the N-terminus and ends with the C-terminus. So if you remember the structure of amino acids, you have the amino group, you have the central carbon, and then you have the carboxyl group. Now, the N-terminus in this case is this side of the amino acid, so where the nitrogen is. That's why it's called N-terminus. The C-terminus is where the carboxyl group, where the C or the carbon is. This is why it's called C-terminus. Now, what this means is that the first amino acid produced will have a free N-terminus, so this won't be connected to any other amino acids, but this will. That's the first amino acid. The last amino acid produced, this will be attached to the next amino acid, and this will be free. That's what it means. It starts from here, from the N-terminus, and ends with the C-terminus. And that's usually the case. Maybe it's not always though, but it's usually the case. Proteins which are meant to stay within the cytoplasm are usually made in the cytoplasm, where proteins which are meant to be exported or transferred to non-cytoplasmic phases are usually made in the rough ER. This is connected to this point that I talked about here. So if a protein is going to be used within the cytoplasm, let's say it's an enzyme that's going to work in the cytoplasm, or it's a signaling protein, for example, that's going to work within the cytoplasm. So it won't be going outside the cell or go inside one of the organelles. Then it's usually finished completely in the cytoplasm. So the translation process starts in the cytoplasm and finishes in the cytoplasm. If the protein is being, for example, transferred outside the cell or exported outside the cell, if the protein is exported to non-cytoplasmic phases, for example, inside one of the organelles, then it's usually made in the rough endoplasmic reticulum because you need to export. And in this case, you need vesicles. Proteins made in the rough endoplasmic reticulum are exported via vesicles, as I said before. So, the first type of translation, as I said, the cytoplasmic translation, or the first example of translation is the cytoplasmic translation. It's when the process of translation starts in the cytoplasm and finishes completely in the cytoplasm. Okay, that's one of the pathways that I talked about. The other one is the one we're gonna talk about in the next few slides, which is cotranslational translocation. So it's using the endoplasmic reticulum. But this is a simple example for the cytoplasmic translation. So this is ribosome, and it's clearly not accurate, actually, representation, but you get the point. Ribosomes, they produce proteins entirely in the cytoplasm. Now, this protein is definitely going to be used within the cytoplasm. So it could be used as an enzyme. It could be used as a signaling molecule. It could attach to the cell membrane, for example, to the inner leaflet of the cell membrane. And in this case, usually through interaction. So if a protein is only made just like this, without any modifications, and it's made for the cell membrane, then it's usually attached to the cell membrane through interactions or chemical interactions. For example, the positively charged amino acids could be attracted to the negatively charged phospholipids, like phosphatidylinositol or phosphatidylserine, for example. I talked about these different examples in a previous video. So this is the first pathway in which a translation is completed, cytoplasmic translation. Starts in the cytoplasm, finishes in the cytoplasm. The second pathway is the rough endoplasmic reticulum translation, or the cotranslational translocation. So this is when the translation starts in the cytoplasm, but finishes in the endoplasmic reticulum. Okay? Now, if you look here, we have constant steps. So these steps, they happen almost every single time a translation happens. And then we have a step that has multiple outcomes, which is step 5 in this case. So if we start from here, when a ribosome starts producing proteins, there will always be a signal sequence. The signal sequence is like let's give you another example, or simple example. It's like a location that is produced by a customer. So let's assume that this protein is a customer. This is the location, and then we have the Uber driver comes all the way and pulls the protein ribosome complex to the endoplasmic reticulum. Okay, so basically, again, first the protein is produced. When the protein is produced, we will have definitely a signal sequence. If there is no signal sequence, then the whole process will continue and finishes completely in the cytoplasm, like the cytoplasmic translation that I just talked about. But for cotranslational translocation, there will always be a signal. Once the signal produced, another protein called signal recognition particle, SRP, will attach itself to the signal and pull the entire complex, the protein, mRNA, ribosome complex, to the endoplasmic reticulum. Okay, that's why it's cotranslational translocation. Now here, at destination, the SRP will bind to SRP receptor here. So this will basically make sure that the whole complex is attached properly to the membrane of the endoplasmic reticulum. Here we have a transleucon. Transleucon is basically a hole that opens for the ribosome and the protein, so protein can be produced in the endoplasmic reticulum lumen. That's what transleucon is. When the whole process is finished, the transleucon will move, basically. This is step 3 and 4. Step number 5, as I said, has multiple outcomes we will talk about in a few seconds. And step 6 is when the ribosome leaves after translation. So again, let me give you the simple example. The signal sequence is like location. So once the protein produces the location, the Uber, the SRP, will go, okay, and recognize this location signal, and it will take the whole complex to its destination, which is the ER. Now, let's talk about the multiple outcomes, step number 5. So this is after the ribosome and the protein are attached to the endoplasmic reticulum membrane. So in this case, we have the signal peptide. That's the signal or the location that I just described. So it's the signal that leads the ribosome and the protein and the mRNA complex to the endoplasmic reticulum. It's also called a start transfer signal because it's a signal to the ribosome as well to start transferring amino acids inside the endoplasmic reticulum lumen in this case. Okay, so start transfer meaning, okay, ribosome, start adding amino acids in this side of the endoplasmic reticulum. There is another signal called stop transfer signal meaning, okay, ribosome, no more amino acids here, continue there. That's probably the simplest explanation for these signals. So that's a signal peptide. It's a peptide that basically leads the whole complex, the protein, ribosome, mRNA complex, to the endoplasmic reticulum. And it's also a start transfer signal, meaning, okay, start adding amino acids in this side of the endoplasmic reticulum. This is a stop transfer signal, meaning, okay, stop adding amino acids here, add them there. This is what basically these signals mean. Now at the end here, you can see that this signal is cleaved through an enzyme called signal peptidase. Now this is only if the signal is at this nascent chain or the nascent chain. So if the signal is here at only the beginning of the protein. Because sometimes the signal peptide or the start transfer signal is actually in the middle of the protein itself. In this case, it's not cleaved because it's an integral part of the protein. But if it's at the beginning of the protein, then it's usually cleaved, as you can see here. The result is a bitopic integral protein. If you want to know a bitopic integral protein, you can check my previous videos. Now you can see that the start of translation started with the N-terminus, meaning that the amino group here is free, not attached to any amino acids. And the C-terminus means that the amino or the carboxyl group here is free and not attached to any other amino acids. Now, one last observation. Inside the ER is outside the cell or non-cytoplasmic phase. So what this means, if this is a protein that is made for the cell membrane, then this will face the outside of the cell and this will face the cytoplasm. Now, let's look at another outcome of this step, which is step 5. Now, similarly here, we have a signal peptide or a start transfer signal, which means that, okay, signal peptide which means basically that the whole complex should be pulled to the endoplasmic reticulum, and that the ribosome should add amino acids on this side. So it starts adding until the whole mRNA is finished. Now, we don't have a stop transfer signal here because the mRNA actually finished. So the whole different codons, or the entire messenger RNA, was being translated or was finished at being translated without producing any stop signal. So you don't need a stop signal in this case. Okay, but you will always need a start transfer signal or a signal peptide for the cotranslational translocation. Because without this, you cannot go from the cytoplasm to the ER, or endoplasmic reticulum. But the stop is not necessary. Stop transfer signal, it's not in every single protein. Now here you can see that the signal peptidase is present. It cleaved this because it's at the beginning of the protein. Again, if it's at the beginning of the protein, if the signal peptide is at the beginning of the protein, then it's usually not an integral part of the protein. That's why it's cleaved, as you can see here. And just to confirm, inside the ER is outside the cell. So this will basically be either exported completely outside the cell, because it's inside the ER, or it will be transferred to one of the organelles, like the mitochondria, Golgi body, etc. Now, the third outcome, possible outcome of the cotranslational translocation is this one. So here you can see that we have a start transfer signal or signal peptide that is in the middle of the protein. It's not here like the other ones. So what this means is the ribosome started producing all these chains, and then suddenly this appeared, and then the ribosome protein complex were transferred to the ER. Okay? So this is a start transfer, meaning also that, okay, add amino acids on this side until the entire mRNA was finished being translated. So no stop transfer signal in this case. Now, as you can see, the end product is a bitopic integral protein, which is similar to the first one that we talked about, but without the cleaving in this case, because it's in the middle, so it won't be cleaved. It will be cleaved only if it's at the beginning. But here, it's an integral part of the protein. Now, another possible outcome for step number 5 in cotranslational translocation is this one. So here, we have a start transfer signal, or a signal protein, or a signal peptide, sorry. So after the signal appeared, the ribosome protein complex was transferred here. And this is also, as I said, a start transfer signal, meaning add more amino acids in the side ribosomes, or the ribosome. And then we have a stop transfer signal here, which means, okay, stop adding amino acids on this side and start adding on this side. And that's what happened until the mRNA was actually finished being translated. Okay, so the result is a polytopic integral protein. It penetrates twice. And again, inside the ER is outside. So if this is a protein that is made for the cell membrane, then this would face the outside, and this faces the inside of the cell, or the cytoplasm. If this is made for the membrane of the mitochondria, then this will face the inner side of the mitochondria, and this will face the cytoplasm, or the outer side of one of the membranes of the mitochondria. Okay? Here, again, there's no cleaving because the signal is in the middle of the protein itself. It's an integral part of the protein. It's not at the beginning. Now, the last possible outcome that I'm going to talk about is this one. So here, again, similar process. The ribosome started translating, so produced all of this part, and then suddenly the signal peptide appears. The complex, the protein ribosome mRNA complex, was then transferred to the endoplasmic reticulum. And this is, again, it's not only a signal peptide, but it's also a start transfer signal, which means start transferring amino acids on this side. And then we had suddenly a stop transfer signal. So meaning, okay, ribosome, do not add any amino acids here, add here on this side. And this is what happened. And then suddenly another start transfer signal. So stop here, start here again. And this is what happens. So basically you could have multiple start and stop transfer signals until the end product, until the whole messenger RNA was translated. In this case, you have 6 parts or 6 basically penetrations of the membrane of the endoplasmic reticulum. And this is called a polytopic integral protein. So if we look here, we start with the N-terminus. So that's a start, maybe. Yes, it's definitely a start transfer signal. Stop, start, stop, start, stop, until the entire messenger RNA was finished being translated. Now again, the inside of the ER, so this will face either the outside of the cell. If this is for the cell membrane, then this will face the outside of the cell, and this will face the cytoplasm, or the inside of the cell. If this is for one of the organelles, then this will face the inside of the organelles, and this faces the cytoplasm, etc. Now, let's talk about post-translational modification. So after translation, sometimes proteins go through processes of modification where things are added to these proteins. Now, the main reason for these modifications could be to target specific organelles, for example, like in the case of the cell membrane. So, for example, lipids or carbs could be added to ensure that these proteins stick to the cell membrane firmly. These modifications could take place in the cytoplasm. It could take place in the rough endoplasmic reticulum or other organelles. We're going to talk about these in a sec. Okay, so this is one of the examples for post-translational modification, which happens in the cytoplasm. So this case, the cytoplasmic translation, so after the whole protein was translated into cytoplasm, there were some lipids added to this protein. Okay, these lipids could be a prenyl group, it could be a myristic acid or a palmitic acid. All of these could be added to the protein. Now, what's the point of this? This is to make sure that the protein is attached to either the cell membrane or one of the membranes of the organelles. Because the majority of a membrane, either the cell membrane or the membranes of the organelles, is hydrophobic, so it's water-hating. Therefore, you need hydrophobic molecules to make sure that this protein attaches itself firmly to this membrane. So in this case, let's assume that this is for the cell membrane, a prenyl group maybe, or as I said, myristic acid or palmitic acid was added to the protein. This protein will go all the way to the inner leaflet of the cell membrane and attaches itself firmly there. Okay, in this case, it's referred to as lipid-anchored protein. And I talked about this in more details in a previous video. So another possible modification could happen or take place in the rough endoplasmic reticulum. So in this case, if you look here, we have this protein that was completely translated in the endoplasmic reticulum. And then we have these different molecules, in this case, or compounds. In this case, we have ethanolamine, phosphate, and then we have phosphatidyl inositol, then we have oligosaccharide. All together are referred to as GPI, or glycosyl phosphatidyl inositol. Now, this GPI is attached to the protein after translation, and this makes sure that the protein is attached firmly to the membrane of the endoplasmic reticulum. In this case, this is most likely transferred or attached to the cell membrane. So this protein is most likely made for the cell membrane because it will be attached to the outer leaflet of the cell membrane. Because as I said before, inside the ER is outside the cell or non-cytoplasmic phases. In this case, as I said, this is most likely synthesized for the outer leaflet of the cell membrane, as we're going to see in the example next slide. So here are examples of post-translationally modified proteins. So here we have the RAS protein, which is most likely synthesized in the cytoplasm, the same with the SRC protein. So these are peripheral protein. So if you want to know the difference between peripheral and integral proteins, then I advise you to watch the previous videos. And these were modified by the addition of lipids, as you can see here. So after their translation, enzymes or lipids were added through the use of enzymes, and these lipids made sure that these proteins stick to the inner leaflet of the cell membrane. In this case, the cytoplasmic leaflet, or the inner leaflet, as I said. So these are most likely made in the cytoplasm. This, on the other hand, is definitely made in the endoplasmic reticulum, as I said in the example previously. So the Thi1 protein, it's an integral, meaning that it will always be attached to the cell membrane, where these peripheral means that they're temporarily attached to the cell membrane. This one was, as I said, definitely made inside the endoplasmic reticulum, and this is a GPI, a glycosyl phosphatidyl inositol. So these are lipid-anchored proteins, which are made in different parts of the cell for the cell membrane, and they were modified after translation. So this concludes our video for today. If you like the video and the content of this video and this channel, then please like, share, and subscribe. You may also visit our website for further updates in the future. Thank you very much, and see you in future videos.